Secretary of Soil Department, Dr. Kurniawan Sigit Wicaksono, Honorable Head of Soil and Water Management Master Program, also our moderator, Dr. Reni Ustiati, SPMP, Honorable our speaker from the University of Chinese Academy of Sciences, Professor Jiansu Wang, Honorable Lecturers of Soil Department, also all of the participants today. First of all, let's praise to Allah Subhanahu wa Ta'ala who have given His mercy and blessed for us. So today we can join and listen to the great speaker on this hybrid event, 3-in-1 lecture, Remediation of Heavy Metal Contamination Soil by Soil and Water Management Master Program. Well, to take advantage of the time, let's begin today's agenda by reciting Basmala together.
Okay, thank you. Can you hear me clearly?
Not yet, sir. Sorry.
Yes.
Can you hear me?
Hold
Can you
on.
hear me?
Yeah, but
So you can hear me, right?
yeah, we can hear you clearly
Okay.
here. But we
Okay.
will have speech from the Secretary of Soil Science Department.
Okay. So I will start, right?
Not yet. After speech from the Secretary of Soil Science Department, please.
Okay, sorry. Okay, sorry. Yes. Okay.
Step on to the following agenda is speech and opening by our Secretary of Soil Department to Dr. Kurniawan Sigit Wicaksono, SPMSC. Time is yours.
Oke, terima kasih Bu Ingar. Thank you. Good afternoon.
Good
everyone.
afternoon. Good afternoon.
Okay. Morning or night or afternoon in your area, Prof. Wang. Let me introduce. My name is Sigit as a Secretary Department of Soil Science. It's a pleasure to welcome everyone to this special guest lecture on the theme of remediation of heavy metal polluted soil, as shown your PowerPoint. Yeah. And this is the vital topic, important topic, as heavy metal contamination process serious risk to soil health, I think, and food safety, and ultimately human well-being. Addresses this challenge, call for innovative, scientifically driven solution, and today we have the privilege exploring same-day approach. Our esteemed guest speaker today is leading expert in the field of soil remediation, I think, is you, Prof. Wang. And we are excited to learn from the wealth knowledge and experience. As a future scientist, engineer, and environmentalist, you will play a crucial role in creating sustainable practice that protect our environmental and promote human health. I encourage each of you as present here to fully engage into the session, ask questions, share idea, and absorb much as you can. Thanks for you being here and making this event a priority. Let us take this opportunity to expand our understanding and inspire the action toward a safer, cleaner, and more sustainable future. Thank you and welcome. Okay, I think it's all. Thank you. Saya kembalikan ke moderator.
Okay, thank you. Thanks for the opening speech. Thank you. It's my great pleasure to meet you online. Okay. So should I start?
Mas Reni, halo. Oke, silakan. Sudah.
Let me introduce our speaker today. I will share screen the curriculum vitae of Prof. Wang. Prof. Wang, could you please stop there first, then I can share screen. Okay. Hold on, wait a minute. So our guest lecturer today is Professor Jiansu Wang. He is from the University of Chinese Academy of Sciences, Department State Key Laboratory of Environmental Geochemistry. You can contact him by emailing him to wangjiansu@vip.ui.edu. GYIG at ACN, and his professional is related to the environmental science geochemistry. His skill and expertise, he is working on the remediation of metalloids such as alkoxyl elements, contaminated environments, and interested in using spectral techniques to enlighten how the toxic effects of contaminants are mitigated, such as contaminants such as mercury, then topic related to remediation, ecological risk assessment, bioaccumulation of heavy metal speciation, and phytoremediation. His discipline related to the environmental science and geochemistry. For his experience since January 2013 until now, he is professor in the University Chinese Academy of Sciences. So related to the project assignment that we post this week about remediation, you can ask further to Professor James Lam. That's all for our guest lecturer today.
Okay, so
You
so
can start, Prof. Wang.
okay, thanks, Rennie. And firstly, I would like to thank Dr. Rennie Astiatic to invite me to give this talk. And actually, it's my great pleasure to have this talk. And I also have a chance to meet David, his old friends. Okay, so I will start my talk. So is my words clear?
Yes.
Okay, perfect. So my talk is about remediation of heavy metal polluted soil. Okay. So my talk includes three parts. The first part is I will briefly introduce why we focus on soil. And the second part, and I will briefly introduce the common, or I would say traditional method used to remediate heavy metal polluted site. The third part is actually I was currently working on this aspect, works on remediation of heavy metal polluted farmlands.
You
Yes,
haven't share your screen.
sorry.
The screen, we could not see your screen.
You cannot see my screen? Okay,
Yes.
I will check.
Please.
Okay, I will check. Share screen.
Yeah, please share screen again.
Okay. How about this?
Yeah, perfect. Now we can see your screen.
Okay, sorry, got a mistake. Okay, so I will continue. So my talk will have three parts. First, introduction. Second is about a brief introduction of remediation of heavy metal polluted site. The third part is about remediation of heavy metal polluted farmland. So I focus on farmland, not site. So every people know this. This is our planet. We can see very beautiful. And we have the huge population, about 7.8 billion people. But for each person, we only own about 5,400 meter squares of habitable land. Even though,
See, Prof. Wang, the screen not moving. I mean, still in the first slide of your presentation.
okay, probably is okay. So I will be a bit slowly. Probably the internet is not so fast. Okay. Let me check.
Please kindly
So
slide.
I will share again. Yes. Okay.
Yeah. You need to
Slowly.
slide show to make it bigger because here
Bigger?
we
Yes, actually, I already is the biggest on my computer. I will
but
check.
we could not see that still showing before slideshow.
Okay. I'll try it again. Share screen. It works?
Yeah, please clicking the slideshow button or by pushing the F5
It
button.
works? No, it works. Actually, it is
No.
not. I will check.
Not working.
Okay.
You need to share it all.
Yes.
Di screen notification.
Yes.
Ini aku nggak
Yes.
share screen. Aplikasinya bukan share screen.
Okay. When you clicking the share screen menu, please
Yes,
kindly choose the share for all.
Yes, I already did, and it seems it works. Did it works?
No, still the same.
Oh. Because here we'll do it again.
Yeah. When you click share screen, then you click the file that you want to present.
Yes.
Then
Yes.
yeah, share for all.
Okay, I will try this one. It works? No? It doesn't work?
Jadi biasanya opsi share screen. Mungkin nggak apa-apa.
Nggak apa-apa ya?
Kayaknya kesulitan pakai screen.
It works?
Oh, okay.
Not yet. Not yet?
Not yet, but we can see your screen now.
Can you see my screen?
Yeah.
Can you see my slides?
Yeah,
No.
now in the slide, our planet. Slide
Okay,
3.
it works, right?
Slide number. Yes.
Okay, sorry. This is software because here we didn't often use this software. So I will continue. Actually, why we focus on land is the second. The reason is although we have like each person in our planet on about 5,400 meters squares have land, but among this land, only about 46% they are used for agriculture. So actually, land is a non-renewable source and is quite precious, especially for 1 centimeters of soil, it will take more than 200 years to produce. So once the soil is lost or contaminated or degraded, so it cannot renewable again. So unfortunately, the United Nations did a survey and they report that about 40% of our land are degraded. And this rise a damage. That means they put ability to fit planet growing population at risk. We know that the degradation of land includes many parts. One of the important part is pollution of soil, especially the human activities like industry and the mining activities. We know that the mining activities will produce wastewater, and this waste also, besides wastewater, it also produce like a mine waste. All they contain like pollutant, like heavy metals. they will, once they discharge into the environment, they will cause pollution. Also, like in industrial activities, they will emit a lot of gases. They also contain the heavy metals. Once the heavy metal deposit to soil, they will also cause contamination. Actually, the soil contamination is unlike soil air pollution. Once air or water polluted, you can see it, you can smell it, you know it is polluted. But for soil, it is not that easy. So the soil pollution has its own characteristics. The first is accumulative. This means that once heavy metals enters into soil, it will be persistent in soil. The second is heterogeneity. This means that when we take soil samples for analysis of heavy metals, even for a same land, if you take the soil samples from different sampling points, you will get the different level of the heavy metal content. The third is irreversibility. That means once soil is contaminated, it will never be cleaned again as it before. The fourth is invariability. So we cannot see it, especially heavy metals enters into soil. You cannot see it. It has no smells. It has no colors. So it is not easy to discover it. The fifth part is difficultiness. So because it is it has invariability, it has heterogeneity, it's not easy to remediate. So soil pollution attracts the global concern, like the Food and Agriculture Organization of the United Nations. They released a report in 2018. In this report, they said about 24 billion tons of healthy soils lost every year due to the pollution. Like in Australia, about 80,000 sites are polluted. Also in China, like in my country, about 20% of the land are polluted. Also in European Union and the Western Balkans, they have over 3 million potentially polluted sites. Also in US, there are more than 1,300 sites. They are on the national priority pollution control list. So actually, soil pollution affects the survival and sustainable development of human beings. So what should we do with the pollution of soil? Should we keep it as it is, or we just abandon it, we don't use it anymore? Actually, many scientists, they propose that we must restore the degraded farmland to feed future generations and protect landscapes. This arises actually a scientific question, is how to use the degraded farmland in a sustainable way. So let's check the common method. If we can use the common remediation techniques or method to restore, to remediate those polluted farmland. Actually, there are many physical-chemical remediation techniques like tilling, soil replacement, soil washing. The tilling probably is the most used method to remediate heavy metal polluted soil, because most heavy metals, they are distributed or concentrated on the top layer of soil. So by tilling, the top layer soil is mixed with the deep soil. We know that deep soil is not polluted, it's unpolluted. So by this mixing, the heavy metal concentration in soil will be decreased. Second soil is replacement. We use a clean soil to replace the polluted soil. But we should be careful that the polluted soil need to be treated properly, but also soil washing is just to use chemicals to wash soil. The pollutant will leach out with the chemicals. Also, the thermal treatment just heating soil to evaporate the pollutant, and this technique is mainly used for mercury treatment. You can see from these techniques, either they need energy, or probably they will cause the secondary pollution, like soil washing, because you use chemicals to wash soil. It will introduce the new environment problems.
Excuse me, Prof. Wang, could you please send your file and I will help you to share the slides.
Okay, it's not so it's not goes well, right?
Yeah, because our audience that joining us online through Zoom meeting told us that they could not see the slide
Okay.
moving.
Okay,
So
so
I
I
will
will.
help. Yeah,
Okay,
please
I
send
will.
the file
Okay.
through email.
Okay. Sorry,
Yeah.
it's this Zoom. I will. Okay. Okay.
Mas recordingnya saya pause.
Okay, I will see this. So I start. Okay. Okay. Move on. Move on. Keep
This one?
going. Yeah. Keep going. Yes. Keep going. Keep going. Keep going. Yeah. Keep going. Keep going. Yeah. Yes. Yeah. Here. Yes. Okay. So I will start. Can you hear me?
Yeah, please.
Okay.
Yeah.
Yeah, sorry. Thanks, Rennie, for deal with this problem. And I will continue. Yes, for traditional method like used for we call the polluted site, not for farmland, it usually has a relatively high cost. Like soil washing, it costs about 75 to 200 US dollars per ton. Also, like thermal treatment, it costs even higher, about 200 to 15,000 US dollars per ton. Like soil replacement, it also costs about 100 and 500 US dollars per ton. So next. So it is a bigger challenge to use those techniques to remediate farmland. So when we use to remediate farmland, the technologies should be capable of. It should be able to remediate large scale of the farmland, because farmland, like in China, our government said that about 16% of the land was polluted. So it's really huge. The second is you must treat in situ. You cannot take the soil out and transfer to another site and to do the treatment. The third part is about the technologies must be less destructive, no secondary pollution. The fourth is lower cost. According to our surgery, we usually accept if a technique it costs if less than 900 US dollars per hectare to remediate farmland is acceptable. So, but this, this is quite for 1 hectare, that's really a huge area of farmland. Also, the techniques must reduce the heavy metal concentration in the edible section of the crops, and it should decrease to an acceptable level. That means it should be safe to human beings. Okay, next. Okay, next slide. Okay. So next, for this part, I would like to introduce the work which I have done for past 15 years. So in my group, especially me, I mainly focus on mercury. So firstly, I would like to talk about some basic information about mercury. So mercury is toxic and non-essential metals. Sorry, previous one, sorry. Especially methyl mercury, it is more toxic than inorganic mercury. Probably you may know the Minamata disease. It is a neurological syndrome caused by severely acute methyl mercury poisoning. And it is reported that the consequence of Minamata disease caused almost 3,000 people affected, and about 1,700 people died. Next, thanks. So our
Next
group,
slide.
yes. So our group did a lot of research at mercury mining site. And we did a comprehensive investigation of the ability of different crops to accumulate mercury from soil. We found that rice can accumulate higher methyl mercury than other crops. And the concentration of the methyl mercury in rice grain is higher than maximum allowable mercury concentration in grain defined by our government. So rice is a potential methyl mercury accumulator. Next. Please move to next. So our group further did work. They investigate. Sorry, previous one. Sorry. Yes, thanks. Yes, our group they study the methyl mercury contained in human hairs in people that both in mercury mining region and in control region, and found that the methyl mercury concentration in here in people living in mercury mining region is higher than control region. Next. Please move to next. And the further study found that the methyl, sorry, previous.
Okay.
Previous one. Yes,
Yeah.
the further study, sorry. Okay, I will continue. So the further study found that the methyl mercury intake from rice positively correlated with the concentration of methyl mercury in the hair. This means that the consumption of the rice causes the accumulation of the methyl mercury in the hair of human beings. Please go to next slide. Next, thanks. So our group further did the calculate the human exposure levels of the total mercury and the methyl mercury, and found that the people at mercury mining region like Wanshan, they have higher total daily intake of total mercury. But if we further look at the methyl mercury, we find that the people at mercury mining region in China, they have the total daily intake of methyl mercury was higher than US EPA standard. This means that a certain number of population at mercury mining region has higher methyl mercury exposure health risks. So next. Next slide, thanks. Yes. So the mercury in rice is from soil. So how to safely use mercury polluted farmland and to produce safe rice is a key issue that I have been working with for more than 15 years. Next, thanks. Next. So actually, we have worked on three types of the method. The first is change of land use types. The second is in situ immobilization. The third part is planting of low mercury accumulation crop cultivars. The aim of this method is to produce crops which contain less mercury. So next, I will introduce the change of land use types. So we did two kinds of work. The first is to study the geochemical fractions of mercury in different types of land. The second, we studied mobilization and methylation of mercury by land use changes. We know in China, we have the two popular land use types. One is dry land. We grow the corns, we grow the rapeseeds, we grow the vegetables, we grow the wheat. The other is a paddy field. We grow rice. Next slide, thanks. So we use sequential extraction procedure to study the mercury fractions in both paddy field and dry lands. We can see that we call bioavailable fractions like soluble and exchangeable, and also specifically solved fractions. These concentrations are quite low. But if we look at the iron manganese oxide bond fractions, that in dry land, this fraction of mercury is higher than that in paddy field. Especially it is about 1 to 2 orders of magnitude higher than the paddy soil. Next. Okay. So we did a laboratory study. So we flooded the dryland soil to see which factors drives mercury mobilization. And we found a very interesting phenomenon that is the concentration of dissolved iron and the dissolved manganese significantly correlated with the concentration of dissolved mercury. This means that reductive dissolution of iron manganese oxide lead to the mercury mobilization during land use change from dry land to paddy field. So the change of land use from dry land to paddy field could enhance mercury risks in soils. Next. Next slide. Okay. So when I was in Germany, I did my postdoc. I was able to use a biogeochemical microcosm to study the mobilization and the methylation of mercury in soil during the redox changes. Because we know that the redox potential of soil increased gradually when paddy field was changed to dry land. So by using this biogeochemical microcosm, we were able to study the mobilization and the methylation of mercury at a wide range of redox potential zones. The left figure is a pH plot, which I obtained by using this biogeochemical microcosm. We can see the lowest EH was lower than -300. The highest EH was higher than the 300 milliwatt. And we take the samples from different redox potential condition. Okay, next. Next, thanks. So this is the total mercury concentration in soil solution at different redox potential conditions. Very clear. We can see that on the low EH conditions, the concentration of the mercury was higher than higher EH conditions. So it seems that high EH favors the immobilization of mercury soil. So this also means that the rising of redox potential could decrease dissolved mercury in soil solution. Okay, next. Next slide. So high EH immobilized mercury. We studied the mechanism that control this immobilization. We collect, yes, so I will continue. So I collect the colloid from the soil solution. From these tubes, we can see it clearly that high EH conditions that soil will produce more colloid in its solution. And under low EH conditions, there's no colloid or collect. So we are curious about the composition, the chemical composition of this colloid. So we use scanning microscope electron spectroscopy to study the chemical composition of this colloid. Next slide. Yes, this is a chemical composition of the colloid. We can see the colloidal particles consist of salts like sodium chloride, clay minerals like silicon oxide, also have iron and manganese oxide. We know that these clay minerals and iron and manganese oxide, they were able to absorb mercury to reduce its mobility. We further studied the colloid bound mercury concentration in soil solution. So next slide. Sorry, go to next. Yes, thanks. Yeah, this is the concentration of the colloid mercury in soil solution. We can see the distribution of colloid mercury concentration was very similar with the presence of the colloid. On the high EH conditions, the colloid mercury concentration was higher. Also, I collect small colloid. So it's quite clear that the absorption of the mercury by colloid decreased dissolved mercury concentration in soil solution. Next slide. So we also analyze the methyl mercury concentration in soil solution. The methyl mercury shows a very similar pattern with the total mercury. And under low EH conditions, the methyl mercury concentration was higher, and it decreased as a function of the redox potential. And on the high redox conditions like 200 or 300 millivolts, the concentration of the methyl mercury was quite low. So we further studied the mechanism that control the decrease of the methyl mercury. Next slides. Next slides. Okay, thank you. Yeah, we know that the methyl mercury is a product of the microorganism activities. Because of the decrease of the mercury bioavailability, it will decrease the methyl mercury production. We also studied the relative abundance of mercury methylation bacteria. We can find that the relative abundance of the mercury methylation bacteria decreased as a function of the redox potential. So the decrease of mercury bioavailability and the mercury methylation bacteria results the decrease of the methyl mercury in soil solution. Next slide. So actually, we know that the paddy field can produce methyl mercury. The dryland has very few methyl mercury. So we did field applications. We did an adjustment. So we changed the land use from paddy field to dryland, and we grow the rape seeds. We can see that before the adjustment, the middle figure, before adjustment, total mercury rice grain was higher, especially this red point. That means the mercury concentration in rice grain exceed the safe limit. Green, that means it's within the safe limit. We can see that the most rice grain samples have the mercury content higher than the safety standard. So we changed these land use types to the dryland. We grow the rape seeds. We can see most rape seeds, they have they accumulate very few mercury. Only few samples accumulate high mercury, exceeding the safety limit. So we did a calculation that before the adjustment for rice grain, the average total mercury concentration in rice grain was 28 nanogram per gram. But when we changed the land use types from paddy field to dryland, the total mercury decreased to 15 nanogram per gram. It's below the national standard for food safety. Also, for methyl mercury, in rice grain, the methyl mercury concentration can reach 16 nanogram per gram. After adjustment to the wrapped seeds, it decreased to 1 nanogram per gram. So by this kind of land use type changes, the mercury pollution risks is well controlled. Okay, next slide. Thanks. So for this part, I would like to introduce what we have done in situ immobilization. So the in situ immobilization is used to spike the amendment to soil to decrease the bioavailability of the mercury. So with selection of the immobilization materials, it should meet some several standards. The first is no hazards to the environment. The second is low cost. The third, the materials are available. We can easily get enough amount of the materials for remediation. The fourth part is no secondary pollution. Next slide, thanks. So with first amendment we focused on is biochar. Probably most of you heard about this kind of material. So biochar is a solid product of thermal decomposition of biomass at a temperature below 900 degrees on the condition of limited oxygen. So the advantages of use biochar is include like environmental friendly. and it benefits for soil. You know, in China, we often use the biochar as a kind of fertilizers to increase the carbon content of soil. Also, it has a potential in carbon sequestration. And also, you know, because of the technologies, we can produce a huge amount of biochars with acceptable cost. Next. Thanks. Next slide. So we used rice husk biochar, and this biochar is we produce in our laboratory. The left figure is the scanning electron microscope image of the biochar. We can clearly see a lot of pore structures of the biochar. And the right figure shows the functional groups, the structure of the carbon of the biochar. And we found that this rice husk biochar was enriched with aromatic carbon. Actually, it's kind of sp2 hybridized carbon structure. Next. Next slide. So we test the different treatment ratios. The first, we spiked 0.6% rice husk biochar. The second, we spiked 3% of rice husk biochar. This is the weight ratio. We can see that after harvest the rice, that's the dry weight of the roots and the above ground tissues of rice significantly increased as compared to control without biochar amendment. These right figures show the rice seedings collected from the different treatment. We can actually, it's quite clear that 3% biochar treatment has a big biomass than other treatment. Okay, next slide. So we monitor it and collect the pore waters of the soil and analyze the mesomercury concentration both in pore water and in soil. You can see that the biochar treatment, both like 0.3% and 0.6 and 3% treatment, their soils contain less mesomercury than control, even in 26 days after transplanting of the rice, 52 days, especially 3%. It is significant. Also, after 87 days, also we saw lower mesomercury concentration in the pore water soil. The right figure is the mesomercury content in the soil solid phase. We can see that it's the biochar treatment also had a very low concentration of the mesomercury. Next slide, thank you. So we are curious how biochar immobilize mercury bioavailability and decrease mesomercury in soil. So we studied the mechanism that control this reduction. Firstly, we monitor the speciation of the sulfur into soil. We use the synchrotron-based techniques to study the sulfur speciation, both in control and the biochar-treated soil. From the right two figures, we can see a very clear differences between the two soils in sulfur speciation. We can see that the biochar treated soil, 3% biochar treatment soil, they have a lot of cysteine. They have a lot of cysteine. Or in control, there's no such kind of sulfur was observed. So I will introduce this mechanism later. Next slide. So we also studied the mercury speciation, both in control and the treated. This means Balta treated soil. There is a clear difference between the mercury speciation, between the control and the Balta treated soil. We can see that there is a significant increase in fractions of the nano mercury sulfate complex. We knew that this nano mercury sulfate complex has very limited bioavailability. This means that the Balta amendment can promote the transformation of the mercury speciation from other forms to nano mercury sulfate. Next slide. Next slide. So based on the sulfur speciation and the mercury speciation data, we proposed a mechanism that control the reduction of the mercury mobility by Balta. So the aromatic carbon structure of the Balta enhance the electron transfer. And this electron transfer promoted sulfate reduction to reduce the sulfur like cysteine, zero valence sulfur, and other sulfates and other reduced sulfur forms. This reduced sulfur, they can react with inorganic mercury to produce the mercury sulfate, nano mercury sulfate complex. This may explain the mechanism that Balta reduce the mobility of the mercury in paddy soil. Okay, next. Next slide. So we analyze the mercury concentration in rice grain, both in control and Balta treatment. It's actually the result was quite optimistic. We can see that 2 Balta treatment decreased the total mercury concentration in rice grain below the national standard for food safety. This means that this Balta treatment can result in the safe production of the rice grain. So next slides. Next slides, thank you. So we also did a field investigation. So we performed a field experiment at the mercury mining region in our province. And we designed a control treatment, a Balta treatment. Actually, the result was similar with the result we obtained from the laboratory. We can find a clear decrease of methyl mercury in pore water, the methyl mercury decrease in rice grain. So these field applications further showed that it is reasonable and it is acceptable to spike the Balta to mercury polluted paddy soil to decrease the mercury content in rice grain. Okay, next. Next, okay. The second amendment I worked on is about carbonate minerals. The reason why I select carbonate mineral, because in our province, especially in southwest of China, we are located at called karst topography. In this figure, it's about the red color, marked with red color. So this carbonate mineral resources, they are abundant in the karst area. So the use of these carbonate minerals has many advantages, like environment friendly, it is kind of clay minerals, low cost, and improve the soil properties. And to enhance the efficiency of the mercury remediation, we used diammonium phosphate to modify the carbonate minerals. So next slide. Next. So firstly we studied the properties of this carbonate minerals. The left figure is the original carbonate minerals without modifications. We can see very clear signal of calcium, carbon, and oxygens. The right figure is the diammonium phosphate modified carbonate minerals. We can see a clear presence of the phosphorus signals. Okay, next. Next slide. So in the laboratory, we performed adsorption experiment. We test the ability of carbonate, the ability of the diammonium phosphate, also diammonium phosphate modified carbonate material. We can see that the solely use, sorry, previous one. Previous, back, yes. We can see that the carbonates, they can use carbonate. It can only remove about 40% of inorganic mercury. For diammonium phosphates, it has very few capacity to remove inorganic mercury from solution. But the modification of the carbonate material with the diammonium phosphate, it can remove about nearly 95% of inorganic mercury from the solution. So this diammonium phosphate modified carbonate material is efficient in immobilization of the inorganic mercury from solution. Next slide. Thanks. So we further studied how mercury was stabilized or immobilized by the diammonium phosphates modified carbonate. After the adsorption experiment, we collect and purified this mercury-loaded precipitate. We firstly use the transmission electron microscope. Use the transmission electron microscope technique to study the chemical compositions of this material. We can see that mercury was associated with calcium, irons, and phosphorus. So we further used the synchrotron-based technique to study which atomics respond with mercury. By using this technique, we found that mercury responded to oxygen and phosphorus, and probably so mercury was likely precipitant as mercury bonded to two oxygens, to phosphorus on the carbonate minerals. Next slide, thank you. Next. So this diammonium phosphate modified carbonate materials showed good performance for mercury immobilization. So we performed the field studies. The first case study was also we did at mercury mining site. The total mercury concentration was about 58 milligram per kilogram. So we spiked this amendment and mixed the soil with this amendment and grow the radish. The case study 2 it's in this study, and the total mercury concentration was 107 milligram per kilogram. We also did the same, and we also tested the radish. Next slide. Next slide, thank you. So we analyzed the mercury fractions, use sequential extractions to see the change of the mercury bioavailabilities before and after the treatment. The initial soil means the soil before the experiment. The control means without spiking of this amendment. The CECM means diammonium phosphate modified carbonate minerals. We can see that the spiking of this amendment decrease the bioavailable fractions of mercury very significantly, both in site 2 and site 1 and site 2. So the result was consistent or in line with our laboratory studies that this amendment can immobilize mercury in soil and in solution. Next slide. Thanks. Next slide. So we analyzed the mercury concentration in the edible section of the radish in control without amendment. The concentration of the mercury was higher than national standard for food safety. But after this treatment, the concentration of mercury below the national standard for food safety. This means that to treat dryland soil, we spiked this diammonium phosphates modified carbonate minerals can reduce the mercury concentration in the edible section crops to acceptable levels. Okay, next slide. So we also developed another amendment we called slow release oxygen fertilizer. Sorry, previous one. Previous. Sorry, back. We called slow release oxygen fertilizer. So we designed or produced this slow release oxygen fertilizer is based on our previous result that increase of redox potential can decrease the mercury mobility. So we thought that our initial thought was to increase the oxygen content of soil. They can create a high EH environment to facilitate the immobilization of the mercury. So this slow release oxygen fertilizer is based on the calcium peroxide. We knew that the reaction between calcium peroxide with water that can produce oxygens. But these reactions can result in strong increase of the pH. Also, they can release the oxygen very fastly. So this is not what we want. So we produce this slow release oxygen fertilizer by using coating layer, fill layer, and buffer layer to neutralize the pH and also to slow down the release of the oxygen. From the left figures, we can see that the slow oxygen fertilizer they can make the pH of the solution not so higher compared to the calcium peroxide. That means that they cannot increase the pH of the environment significantly. Also, the right figure shows that the dissolved oxygen concentration in the solution. We can see that the oxygen fertilizer we produced they can slowly release oxygen and keep it or remain it for a longer time. Okay, next slide. Next. So we spiked this oxygen fertilizer into the paddy soil. We can see that that's the control soil, the left figure. The dissolved oxygen content was quite lower in control soil. But in the oxygen fertilized treatment soil, it can still remain around 4 milligram per liter. Also, the right figure, previous. We can see the redox potential. The control soil has a very low redox potential, but for oxygen fertilized treatment soil, the redox potential remained around 100 millivolt positive. So it reached our initial aim to increase the redox potential of soil. Next slide, thanks. So let's look at the dissolved ions and dissolved manganese in soil solution. We can see that the oxygen fertilizer treatment significantly decreased the amendment of the oxygen fertilizers inhibits the reductive dissolution of iron manganese oxide, and then inhibit the mercury mobilization. So yes, go on. Next, yes. Proceed. Next slide. Sorry, next. So let's look at the dissolved mercury concentration in the pore water. And it's very clear that oxygen fertilized treated soil has rather low mercury concentrations compared to control. This means that the mercury mobilization was inhibited. And this is because of the block of the reductive dissolution of iron manganese oxide. Next, thank you. Next slide. So let's look at the soil methyl mercury. And the methyl mercury shows a very similar trends with the dissolved mercury. The oxygen fertilizer treated soil, we can see the methyl mercury concentration in soil was almost very low, was around 0.12. But in control, it increased to almost 30 ppb. So oxygen fertilizer decreased the methyl mercury significantly. Next slide. Next. Yes, back. Yes, we find a linear correlation between the dissolved mercury and the soil methyl mercury concentration. So this means that the decrease of dissolved mercury lead to the reduction of the soil methyl mercury. This is one of the important mechanisms that control the decrease of the methyl mercury. Okay, next. So we know that the factors affecting the mercury methylation include the bioavailability of mercury, which I already showed in my in the last slides. Sorry, back. And another factor is mercury methylation microorganism. So we studied the relative abundance of the mercury methylation microorganisms use 16S RNase sequencing. Next, thank you. So this figures show the relative abundance of mercury methylation bacteria at genome levels. CK means control without oxygen fertilizer amendment. OF means oxygen fertilizer treatment. We can see a clearly result at 19 days that in the CK control, the relative abundance of mercury methylation bacteria was quite higher. But if we look at the OF, oxygen fertilizer treatment, the relative abundance of this bacteria was quite low. And it's decreased about 97 to 90% in abundance as compared to control. So this oxygen fertilizer was quite efficient in reducing the bioavailability of the mercury and inhibit the colonization or growth of the mercury methylation microorganisms. Next. Next slide. Sorry, next. Yes, we further test the amendment of different amount of this slow release oxygen fertilizer on mercury mobilization and the methylation in soil. Actually, we found that actually spiking of the 1% of this slow release oxygen fertilizer can significantly reduce the both total mercury in the pore water and the methyl mercury concentration in soil. Yes, yes, next. Next, yes, please. So we also test the ability of this slow release oxygen fertilizer on mercury mobilization and methylation in different mercury polluted soil. One soil is was collected from the high geological background soil. The second soil was polluted by mercaptan. The third soil was polluted by the atmospheric mercury. We can see that the amendment of this slow release oxygen fertilizer can reduce all methyl mercury concentration in these soils. It's quite efficient and the result was quite clear. Okay, next. Sorry, next slide. Yeah, this is methyl mercury and next slide, yes, where they did.
Halo, Prof. Wang. Halo, Prof. Wang. Mas Anto di pause dulu recordingnya. Kayaknya beliau. Ya. We can
Okay.
hear your voice. I will share screen again and we can continue
Okay,
our
sorry.
section.
Sorry for this. Yes. Okay, so I will start. Can you hear me?
Yes.
Okay. Actually, okay. So this is done. This is done. Next. Yes. Next slides. Thank you. Yeah, actually, it's another approach or method we used to reach the aim of the safe use of the land is to plant low mercury accumulation crops. Can you hear me?
Yes.
Okay,
Yeah.
go ahead. Okay, please. Okay, yes. Actually, we find that the crops are variable in capacity to accumulate mercury to their edible sections. And so we systematically studied the mercury content in the edible sections of vegetables and fruit and crops at the mercury mining site in China. Also, we studied the ability of different cultivars of rice to accumulate mercury from paddy soils. Next. Yes, the top figure shows that the mercury concentration in the edible sections in vegetables. And we can see that that's the different vegetables, they have accumulate different levels of mercury in their edible sections. The line around the 10 nanogram per gram means the national standard for food safety. So we can see that there are only two vegetables like radish and garlic. They accumulate less mercury and almost reach the national standard food safety levels. Also, because the second figure, the lower figure shows the total mercury concentration in soil. We can see that even especially for the radish or for the garlic, we can see even the total mercury concentration in soil reaches like the 100 ppm. They still accumulate less mercury in the edible sections. So this means that this radish and garlic probably is a kind of vegetable species that have very limited ability to accumulate mercury from soil. So next slide. Next. Okay, this is mercury concentration in the edible section of the fruit. The left figure is mercury in the fruit. The right figure is the mercury concentration in the paired soils. We can see that the soils mercury concentration are quite higher, also reaches like the 100 of the ppm. But a very interesting thing is that we find a few fruits like the watermelon, strawberry, grape, orange, which has a mercury concentration in their edible sections below the national food standard for safety. This means that the growing of these four fruits like orange, grape, strawberry, watermelon, they can safely grow in the mercury polluted soil and accumulate less mercury. Okay, next. Also, this is the crops. This figure shows the mercury concentration in crops collected from the mercury mining site. You can see that the corns and the sweet potatoes, they have the mercury concentration below the national food standard. So these two crops probably the potential low mercury accumulators. Next, thank you. So we did a more detailed study to study the different ability of the rice cultivars to accumulate mercury from soil, because in China we eat a lot of rice, and rice is the main stable food in our daily life. So we test about 60 cultivars, and we did find 2 or 3 cultivars that has very limited ability to accumulate mercury from soil. And the concentration of the mercury in the rice grain almost reach the safe standards. Okay, next. Yeah, we also did a field experiment, and we grow the different rice cultivars, and also find that the growth of the low mercury accumulators, rice cultivars, they can produce rice with very few mercury in the rice grain. Okay, next. We also test the different ability of the different ability of different cultivars of tomatoes, beans. We also did find like some cultivars they accumulate very less mercury in the edible sessions. Okay, so because these are Chinese, I will not talk about their names. Next. Next, yes, this is for chili. We also test the different ability of chili to accumulate mercury. We also did find like these vegetables. We also find a few vegetables that were accumulate very less mercury in their edible sessions. Okay, next. Yeah, this is a chili. Actually, most chilies they accumulate less mercury in their edible sessions, and we did find like several species like the first or second concentration of mercury was very low, so it is recommended to grow at the mercury polluted site. Next. Next, thanks. So we also discovered some low mercury accumulators oil seed wraps. So I will note this is the photos of our field experiment, and we did find they accumulate less mercury in their grains and can grow safely in mercury polluted farmland. Okay, next. So then probably I will go to the almost to the last part of my talk. It's about it's not about mercury. It's about the cadmium, zinc, and lead. So in this part, I will introduce the work. Actually, it was done by some of my friends. They tried to save use of heavy metal polluted farmland, use the combined remediation techniques like grow the low heavy metal accumulation crops, and then meanwhile they spiked the amendment to soil to immobilize the heavy metals. Okay, next. So this is the information of the experiment site. We can see that in the experiment site, the total concentration of the lead, cadmium, and zinc that was higher than the Chinese national standard. Also, the rice grain grow in this soil. They also accumulate a high concentration of the heavy metals. But for lead, it's below the national standard. But for cadmium, most samples have the cadmium concentration higher than the national standard. Okay, next. So this is the general physical chemical properties of the experimental soil. The soil has quite a low pH, about 4.2, and also have the total concentration. Also have the total concentration of cadmium about 0.9, and zinc 246. And also lead is 162. So it's all above the Chinese national standard. Next. So the mass varieties cultivars use three different cultivars. It was defined previously defined as a low heavy metal accumulators like Yunshi 5, Huanong 1, and also Remediation 1. So immobilization agents include lime, because the soil was acidic, I used lime to increase the pH. Also, clay mineral. Also include we called nanosilicon foliar fertilizers. So these figures show the field experiment, the cone, also the treated soil. Next. So this is mercury concentration in the grains of the cones after the first season of after the first season. We can see that for CK, that means the CK that they have no amendment was spiked to soil. That different cultivars of the cones, they accumulate different concentration of zinc and cadmium. But when the soil was treated by lime, increase the pH, the concentration of the zinc in cone grains significantly decreased. Like in the Yunshi 5, the concentration of zinc decreased to 50, and the cadmium decreased to 0.17. All these treatments are significantly result significantly lower zinc and cadmium concentration in the cone grains as compared to control and the other treatments. Okay, go ahead. Okay, next slide. So this is the second year's result. The concentration of the heavy metals in the grain of the cones. So we only show the control CK without lime treatment and lime treatment. We can see that lime treatment results a very lower concentration of cadmium, lead, and zinc in the grain of the cones. Also include copper. Like the second column shows in another season 4, we can see the efficiency was in season 4 was not so efficient as compared to season 3. But overall, that the treatment of the soil with lime combined with low accumulation mass cultivars, they can result a very lower heavy metal concentration in the grain of the cone. Next slide, thanks. So another technique is about photoremediation. In China, we have many companies and many scientists use photoremediation technique to remediate the heavy metals. Next. So we actually in China, we also discovered a number of the heavy metal hype accumulator plant, which were efficient to uptake heavy metals from soil. Next. So in China, a group they did a study. They grow these hype accumulators in heavy metal polluted soil to see ability of this hype accumulator to uptake heavy metals from soils. This figures show the photo. This photos show the field experiment. Okay, next figure. Oh, sorry, next slide. So this is this hyper accumulate the amount, the concentration and the amount of the heavy metals extract by this hyper accumulator. That they calculate the extraction rate. It seems that it seems quite higher. It reaches 60% of cadmium was extracted from soil every year. So actually, this value is higher. But this is not usually accepted for farmland. So please go to next slide. Because although this shows promising and it have a 60% extraction rate for cadmium, but it still is time consuming. The most important it is interruption of the agricultural production. Usually when we talk about the remediation farmland, we mean that we can still grow the crops on those polluted farmland. But if you only use the photo extraction to grow the hyperaccumulators, you cannot grow crops. So this is a problem. Please go to the next slide. So scientists, they have developed we call intercropping system that grow the hyperaccumulators. Also, they grow the crops like maize, like corns, and low accumulation crops. By this way, we can use hyperaccumulator to extract heavy metal from soil, and meanwhile, we grow crops because of its low heavy metal accumulation ability, so it can still produce safe crops. That means that the corn grains accumulate less heavy metals that can meet our national standard. This is a photo of what they did in the field. Okay, next. Next slide. So this is the total concentration of cadmium in soil after 5 harvests. That the blue one, that means they only grow the hyperaccumulators. This and these squares means with this pink, that means intercroppings. They grow the maize, corns, and also with hyperaccumulator. This dark triangle, this means that intercroppings with the corns, the hyperaccumulator, they also spike some chemicals like EDT to enhance the mobility of the heavy metals. We can see that after 5 harvests, the concentration of cadmium decreased from 1.4 to 0.3, and actually it is quite efficient. Let's go to next slide. So let's check the cadmium concentration in the grain of the corn. We can see that at the first and the second harvest, the concentration of the cadmium was higher than the national safety standard. But at the third and the fourth harvest, the concentration of the cadmium was lower than national safety standard. This means that we use the intercropping system to grow the hyperaccumulators with low accumulation heavy metal cultivars crops. We can reduce the total content of heavy metals and meanwhile produce crops which accumulate less heavy metals. It is a quite a promise and optimistic technology used in China. Please move to next slide. Yeah, this is yield. This is the biomass and the grain yield of the different treatment. For this is for corn. We can see that the stem and the leaves, also grains, they produce a significant amount of the biomass. And actually, there's when you spiked some chemical agent like EDT, it can increase the biomass a little bit. But anyhow, this intercropping can produce a significant amount of the biomass and the rice grain. Okay, next slide. So we also actually, besides the intercropping between the maize with the hyperaccumulator, some professors also like to do the intercropping with the hyperaccumulator with some trees like oranges, like already such kind of technique is widely used in Southwest China. Also next.
See it?
Yeah, yes, actually this last slide. And thanks for your attention, especially I use of lines sometimes, and I mean sometimes the voice or the technical problems makes some not so smooth. I hope you can at least get some information from my talk, can get some general idea of how to save use of heavy metal polluted farmland in China. Actually, the method I introduced is the most popular method we use currently in China. Okay, thanks, Rini.
Thank you very much, Prof. Wang, for your insightful talks. Now we are moving forward to the Q&A session.
Okay.
So
If
ask here on
yes,
the
yes, Rini, I mean sometimes the voice was not so clear. So can you put the question into the use this online? Write the questions. Okay, okay.
Okay,
So
also have a participant that joined through Zoom. For participant that wants to ask a question, you can write your question to the comment box in the Zoom.
yes,
And
please do that.
for that want to ask directly, please, you can raise your hand. Okay, please come closer to the microphone. Mention your name, then your question, so Prof. Wang can see your face, also can hear your voice clearly. Okay, we have Michelle. Come closer, Michelle, please. Here.
Yes.
Yeah.
Thank you for the. My name is Michelle, and I want to ask two questions. The first question is, how long does it take to reduce mercury concentration in rice grains after adding biochar to the soil? And the second question, should biochar application be done periodically, and if so, how many applications are required to effectively reduce mercury concentration in rice grains? Thank you.
Yes, I got the first question is about rice, right? Mercury rice. Yes. Rini, can you repeat this question?
Okay. Michelle asks about how long it will take to reduce the methyl mercury concentration in the soil after adding the biochar. And the second question, how many times, like the frequency for adding the biochar to actually cleaning the soil from methyl mercury.
Okay. Okay, thanks for the question. Actually, when we spiked the biochar to soil, then we directly grow the rice on the biochar treated soil. It actually it will take about 1 or 2 months, 1 or 2 months, probably about 2 months. The methyl mercury concentration in soil will decrease significantly. 2 months, and usually we spiked the biochar 1 month earlier, before transplanting the rice, and then we spiked the biochar and flooded the paddy field to make the reaction happens. The biochar's mercury to make the reaction happen, to decrease to make some, to all of the mercury to transform from the other forms to transform to the mercury sulfide. This will take like 1-2 months. Usually it's 2 months. But the spiking of the biochar, we didn't aim to like reduce the content of the mercury in soil, because it cannot remove the mercury from soil. It just affect the mercury speciation, affect the mercury mobility, just decrease the availability of the mercury in soil, and to inhibit the rice uptakes the mercury from soil. I'm not sure if my answer was clear or not.
Yes, Prof. Wang. And the second question about the frequency. How
Sorry, can you
many
repeat?
times? How many times it's required to apply biochar until the soil is clean from methyl mercury?
Sorry, I didn't get you. 3?
Her second question is about the frequency or
Frequency.
what?
Frequency, okay. That means like frequency, that means to spike the biochar, right?
Yeah. Yes.
Actually,
How?
it's we actually based on our current result, it's if you spike the biochar, at the spike the biochar, probably it will like works for probably 2 years. This means that we spiked biochar in 2 years, it's fine, but after 3 years, it seems like you need probably you need to add more biochars. Yes, 2 years is the result was acceptable. Yes, was good. We can see the decrease of the mercury in the crops. But in third year, in third year, its effect was become quite weak. We needed to spike some fresh biochar, to. Okay.
Okay, thank you, Prof. Wang. We have more students that have a question. We have Faiza. Please mention your name and also your question. Faiza.
Okay. Hello, Mr. Wang. Thank you for your
Hello.
opportunity. My name is Faiza Mutiasari. I'm from Water and Soil Management. My name is, and then my question is, I missed the information. How to easily to detect soil if that has been contaminated by heavy metals? And when the soil and the plant have been contaminated, where is the dispersal of biomass?
Okay, I get it. Okay, so Faiza question. The first question is how to easily detect that the soil is contaminated with heavy metals, like a rapid judgment that the soil is contaminated with heavy metals. And the second question is when we use a plant as accumulator of heavy metals, what we can do, how we can dispose the biomass that contain heavy metals inside
Okay.
the plant. That's
Okay, thank
yeah.
you. So firstly, I would like to answer the second question. The disposal of the biomass of this hype accumulator. Actually, in China, we have did a lot of work on this, this photo extraction, photo remediation techniques. I like some of my friends, of my colleagues, not in my institution, they developed a kind of facilities. They can treat the biomass of the hype accumulators. First they burn it, they collect the ash. They collect the ash of this biomass, and then they treat it as a kind of solid waste. This actually this is quite, we did a lot to treat the biomass of the hyper accumulator in China, especially but not in our institution, in another universities. Sorry, the first question is about the detection of the methylmercury.
Contaminated soil, like a rapid detection.
How to detect the methylmercury, right?
Yeah.
Ah,
Like, how we can detect, how can we know that the soil is contaminated with heavy metal?
Okay, okay. Actually, in China, we have a national standard for the maximum allowable heavy metal concentration in soil. So usually we take soil samples and analyze the heavy metal concentration in soil samples. If these values were higher than the standard, this means that they are polluted. They need treatment. They need remediation. So we have a very clear standard. Like for mercury, it's about 3.6 ppm. If it's higher than 3.6, the government needed to do something to treat this polluted farmland properly, to make sure that the crops can grow safely. Okay.
Okay, Prof. So we can know that the soil is contaminated with heavy metals by doing analysis. But Faiza still curious whether the soil that contaminated with heavy metals, like showing certain sign or symptom that the soil is contain high concentration of heavy metals, like for the physical properties or biological properties.
Sorry, I'm not clear. I'm not. I get a few.
Okay, let me repeat again.
Okay.
Faiza is still curious whether we can have like how to say it, like rapid judgment without doing analysis in the laboratory, that this soil, by showing the soil, we can see that this soil is contaminated with heavy metals. Is there any
Yes.
certain sign that we can see by like naked eye, this soil is contaminated with heavy metal,
Okay.
something like
Okay. I got your question. Yes. Okay. Actually, it's very difficult to see by the eyes. But usually without this chemical analysis, we can also judge. We can also get some information whether this soil was contaminated or not. The first is to see if the soil located close to a factory or close to like a mining site. If it is close to such facilities, it's more or less it will probably it may be contaminated. If it close to a mining site, also we can get some information about to ask some people, ask the landowners if he frequently use some pesticides, like chemical fertilizers. If used too often, that means probably it may contain some heavy metals. Yeah, but it's difficult to like just see or smell. It's it's not a it's actually it's not very difficult to see it whether it is contaminant or not.
Okay. We still have a student that have question. Arif. Okay, Arif. Please mention your name and your question.
Okay,
He is Arif.
thanks for the
Louder,
opportunity.
please.
My name is Arif Pravan. I have two questions. And the first, it's maybe continue the Michelle question. How many doses of RHP or the biochar application in tons per hectare so it can effectively decrease the mercury? And the second, what is the difference when the farmer directly burn the residue from the paddy compared to the application of biochar to decrease the mercury? I think it's same. The both is same. Burn. Yes, it's burn.
Lorena, I need your help.
Yes.
Okay.
So,
Let me repeat. Just
sorry, it's not so clear because it's a little bit
Okay,
far
yeah.
away.
Yeah, because Arif sitting far away from the speaker also. His first question is about the dose of RHB, rice hull biochar, that can effectively decrease the methyl mercury concentration in the field. And the second question is compare to apply RHB with the directly burning the rice hull without making biochar. Is there any difference? Because the mechanism is same, like burning biomass, but for biochar it called as pyrolysis, but directly burning it's like a different temperature or something. And his concern is there any difference? Because the method is the same, like burning the biomass.
Okay, so it's so I will repeat the question if I got the question correct or not. So his question is about its application of the biochar, right? IHB biochar. It's about its high temperature, like pyrolysis of the biomass, right? So he's
let me repeat again. His first question is, what is the dose of rice hull biochar that can effectively decrease the methyl mercury in the field? The dose, like how many tons per hectare that can effectively reduce the methyl mercury in the field. And the second question is, in Indonesia, we have many farmers that apply like the burning ash of biomass to the field. And in your research that you present previously, you said that the rice hull is burned through pyrolysis to make a biochar. So what is the difference between applying the burning biomass ash directly compared to make the rice hull biochar first?
Okay,
That's,
thanks. Yes, I would like to answer the first question about the amount of biochar used to treat soil, right? Actually, we calculate the suitable amount of which can effectively reduce the methyl mercury rice grain is around 24 tons per hectare. 24 tons per hectare. 24. Around 24. And for second question, what's the differences between the biochar and you often observed that the burning of the biomass, the burning of the biomass in the field? The biochar is produced under the limited oxygen conditions, but what you saw in the field, it is burning in the air. If you burn those biomass, in a container or something, probably you may also produce biochar. Because those things were spun in the air, so it will completely burned, to commonized. But for biochar, we produce, very few oxygen inside, so the most biomass will transformed, will be converted to carbon structures. But for normal, like the air, it will totally consumed, we will totally burnt, and you get some ash. So if we want to get biochar, probably you need to put those biomass in a container, and only allow a few oxygen to come in. Then you will also get biochar. Actually, biochar is very easy to produce. Very easy.
Okay. Any feedback, Arif?
Enough.
Enough.
Thanks, Prof. Wang.
Okay, thanks, Prof. Wang. And we still have many questions from our student, but we will give a chance for a student that joining us online. We have Wirda Azahra who raised her hands. Please proceed your question.
Okay.
Okay, thank you very much for Dr. Reni for change. Okay, the first I want to say thank you for Professor Wang for the lecture today. It's very interesting, and also I want to say thank you for Dr. Reni to provide this lecture today. So I hope Prof Wang and Dr. Reni you are doing well now. Because in
Yes.
Beijing now is very cold.
Oh, really? Okay.
Yes, it's very cold for me, and maybe it's not usual for me. Okay. So my questions, Professor, as mentioned before in your presentation, the strategy or the ways to control the mercury in the soil. The first is change the land use. For the example, for your research, like after adjustment paddy land to dry land, it was effectively control methyl mercury. But I'm still confused because I ever read some journal or article that mention if in the dry land, the process of the decomposition of organic matter more faster in the paddy land because in the dry land because in the dry land the activity of microorganism in the soil will increase in an aerobic condition. So it's mean that the amount of the organic matter will decrease. So which one is the better way to just keep the paddy lands, or we still change the paddy land to the dry land to control the methyl mercury? The first question, and then the second question is about as I mentioned it before, that in the paddy land to dry land, your research planned the rapeseed to control mercury. So my question is, are there any other crops that are able to control methyl mercury, or the use of rapeseed in the plant cultivation the most effective compared with the other crops? Okay. That's my
Okay.
question. I mean,
Okay.
like, it's any other crops to control between rapeseed and other crops to control mercury in the soil. Yes. That's
Okay.
my
Okay.
question,
Okay.
Professor.
So
Thank you very much.
okay, the first question, actually, the first question is about this change of land use. Actually, in China, we have a very strict policy. If the soil was contaminated, exceed our national standard, that you are not allowed to grow the crops in those land. This is very strict. But if you have some solutions, you can reduce the mercury concentration in the crops which grow in those heavily contaminated soil. to below our national standard, then you can use those soil. So like in mercury polluted site, especially in mercury mining region, the mercury concentration in soil was extremely higher. They are not allowed to grow crops, especially grow the rice, especially is not allowed for grow rice. So we recommend them to not to grow rice. You can grow the coals or wrapped seeds. After that, because after this change of land use, the mercury concentration in the corns, in wrapped seeds, mostly they are below our national standard, so they can use it. So at the moment, the critical thing is to find a way to make sure whatever crops you grow, you must make sure that crops contain that mercury below our national standard. This is a very critical issue. So at the moment, we only think about to find a way to make sure that crop can grow safely. Like for the soils, organic medicines, fertilities. At the moment, we didn't take care too much about this issue. Oh, okay. This my answer to your second question is about the other crops, right? Other crops that can accept the wrapped seeds, right? Except the rapeseed, you can grow safely,
Yes,
right?
to replace, yes, and maybe there is another crops to we can use to replace the rapeseed maybe in the
Yes,
cultivation.
yes.
Yes.
Yes, actually, well, the most successful practice at the mercury mining regions is currently to grow the fruits similar to oranges. Actually, we actually they grows about like 100 more than 1,000 hectares. All those lands, they are heavily contaminated. They are not allowed to grow the crops. But if they grow this kind of fruits, actually I don't know the English name, but it's similar with oranges. So it's the concentration in these fruits are quite low. So no, they are large areas. They are lands. They are grow such kind of fruit. So they are farmers are they are very happy to do this because they are not allowed to grow crops. They have sometimes the critical issue of the social economy point of view. But when you change the land use to this orange, they can sell in it. They can get some money. So they are very happy. They are keen on to do that. So that's the most I would say the most successful strategies or we use to control the safe use of mercury contaminated land. Okay, this is my answer.
Thank you very much, Prof. Wang. Thank you, Wirda Azahra, for the question, and we still have questions from our student that joining us in this conference room. Please mention your name and your question. We have Ragil.
Good afternoon, Professor Wang. My name is Ragil. So after I check your recent researches, your research mainly the mercury contamination caused by gold mine. But my question is a little different, but still in mercury problem. My question is a mercury that caused by agricultural practices. Such as the application of fertilizer and pesticides. So my question is, have you encountered the mercury contamination caused by the agricultural practices? And if you have, my next question is there a way that that we can extract the mercury contamination more than the mercury intake caused by the fertilizer, because as we know, in Indonesia, especially Indonesia, the most common practices is the intensive agriculture that dependent in the fertilizer. So we cannot stop using the fertilizer. But so is there a way that we can extract the mercury more than the intake from the fertilizer?
Okay. Okay. So,
Yeah.
yes, it's about the mercury contamination in soil by fertilizer, right?
Yes.
Okay. Actually, in China, especially in my province, we didn't care about too much about this fertilizer. But this is only in my province. If we went to the other province in China, they have the same problem because of the application of the fertilizers, pesticides. They result in the mercury contamination in soil. But in my province, it's mainly about these mercury mining regions. We have a lot of mercury minings. We have also high levels of mercury in soils. This is natural resources because of this, these mineralizations, this mercury deposit makes the whole province. They have the high levels of the mercury in soils. So actually, our method is although I present for this highly mercury contaminated soil at mercury mining region, but it's also applies for the mercury for soils farmland contaminated by these fertilizers, because it's especially in China we have a general accepted strategies is to change the cultivars of the rice, to change the species of crops for the mercury contaminated regions, either in mercury mining regions or in a region that soil was polluted by fertilizers. It doesn't matter. You just need to change the cultivars of rice, or use some species, some crops that accumulate very few mercuries. So I'm not sure if my answer was correct or not.
Yeah, it was very clear. Thank you.
Okay. Any question? Okay. Thank you, Prof. Wang, for your insightful talks. Also, respond to our student question. Now we have a little bit sign of our gratitude that you already sparing your tight schedule for giving us our student lecture. I mean, here for the certificate of appreciation. And please give round of applause to Dr.
Okay, thank you.
Thank you very much, and we will send this certificate to your email, and we would like to request take picture together. So, Ragil, please you can come.
Yes.
And please, for all students that joining us online, you can open your camera. Okay. Like a good position. This is already good position. Oh no. It's help me. Okay, Arif. That's only me that only focus. Okay. Can we come closer to this side? Because we have this our values. Excellent.
Okay.
Hey, don't do that. Come closer to this side. Arif, Tama, Acha, and other students, please come closer. Okay. Come closer. And I will take picture from my computer by counting 1, 2, 3. Okay, Acha, you are next. Okay. Okay, Tama. Okay, Hasik, please. Okay. Let me count. 1, 2, 3. Please help me from here. Okay, once more. 1, 2, 3. Hold on, hold on. Okay. Thank you very much, Prof. Wang.
Okay, thank you, thank you all.
Thank you so much, Prof. Wang.
Okay, thank you, Rini, thank you, William, thank you all.
Hope to have another chance in we can hope to meet you in another chance in guest lecturer or international conference. Have a good evening in China. Thank you very much and see
Okay,
you.
thank you.
Bye
Thank you.
bye.
Bye. Bye.
Thank you, Prof. Wang.
Bye.
See you.
See you.